Hello, everybody. Welcome to tonight's Q&A. My name is Jackie Zimmerman. I am a long-term IBD patient, and I am here with some members of the Gali Health team. Tonight, we are talking about their IBD Footprints program. Just a little bit about the who, the why, the what, all of the stuff, the questions surrounding the program itself, and why you should join and why it's important for the IBD community. So before we hop into all those super fun questions, I'd like to introduce these two members of the Gali Health team, Arie and Arielle. Don't, pay attention.
We didn't plan
who we're talking to. But Arie, would you like to tell us a little bit about yourself?
Oh, hi there. I'm Arielle, and I'm heading the discovery effort at Gali Health. I'm the one behind working with the team to design the strategy for the Footprint program.
Arielle, tell us about you. We know you, but tell us again.
My name is Arielle Radin. I'm the Director of Clinical Research at Gali Health. So I'm kind of in charge of trying to get patients, our community members excited and involved in our research efforts.
Arielle is the lady who will send you emails if you forget to send in your sample.
Yes, sometimes I help Gali out with reminders.
So in order to talk about this kind of research, because this kind of research is not the same as a clinical trial, right? We did a chat a couple weeks ago about the differences in research. Would either of you like to just give a real quick, what's the difference between what we're talking about today and like a clinical trial?
I can start. So I think the main difference is that with what we're doing, it's technically an observational longitudinal research study, which means like we're really just observing you. We're not doing anything to you beyond taking your poop. But we're not actually intervening. We're not giving you any medications. We're not trying anything out on you. I know sometimes patients are reluctant to take part in clinical research because they think they're going to be a guinea pig or a lab rat and we're testing stuff out on you. That's not really the case at all. I think observational research is really cool, especially within clinical research, because we get to know the experience, like the actual day-to-day experience of patients, and that helps us inform clinical trials later on. So essentially, if you want to make sure that like eventually one day when they're testing out medications or they're discovering new medications, you want to make sure that your experience with IBD is represented in what goes into developing those types of drugs. Observational research is really cool to take part in.
I think the observational part is really the key takeaway, right? Yeah. You're just watching, right? It's a minimally invasive kind of program to be a part of, right? It's really, really easy to take part in what we're about to talk about tonight.
Yeah, it's really just your time. Yeah, I would say like that's pretty much all you're giving up is time. You're giving up some poop and you're just sharing your experiences with your illness with Gali. That's pretty much what's involved.
Easy peasy. So because we're talking about science tonight and research and all these like super awesome nerd topics, there may be some jargon. So before we dive in, I do want to ask if you guys could help us clarify a couple terms that either might come up tonight or that maybe we've seen in the general sphere of research. The first one I want to ask is one that is, I feel like I see more and more and more, which is biomarkers. What is a biomarker?
Yeah, I mean, the way I think about it is honestly, it's two words put together, right? It's like biological and marker. So it's really some kind of biological phenomenon, whether it be like some component of a cell or like the activity of something going on in your body that can be used as a marker for something like diagnosing someone with an illness. So if you have this X biomarker, it's a good indication that you might have IBD or something. Or it could be a marker of disease activity. So a lot of people know about fecal calprotectin. So getting those tests done, it's a marker of disease activity because it's basically just showing GI specific inflammation. What's cool is that we're also now interested in digital markers. So there are biological markers, which are coming from your body, right? But then there's also what we're thinking about as digital markers, which are essentially these like digital signatures that are coming from the Gali app. So providing any kind of symptom experiences you're having in your health monitors, or even like passive, people are really interested in passive digital markers. So like how many steps did you take this day? That could be a digital marker that you're getting from like a fitness tracker or your iPhone, something like that.
It's kind of like leveling up patient-reported outcomes because it's digital now.
Mm-hmm.
Okay, what about disease signature?
Arie, I feel like they're similar, right? Like they're basically the same, right?
Signature is many biomarkers together, so
Yeah.
that we're measuring several things together, and they somehow they correlated together statistically, pointing out for the disease course. So it could be more than one biomarker. So the simplest way, for example, to really think about biomarker is like cholesterol. So you could imagine that you track your cholesterols over time, and it's a biomarker for predicting cardiovascular diseases. So everybody is going to the doctor once a year to measure the cholesterol just to make sure that the cholesterol is under a certain level to really reduce the risk for cardiovascular disease. Similarly, with IBD, things are a little bit more complicated because it's a very dynamic and very complex disorders, a disease, and it's hard. There's not many biomarkers are available. The only biomarker that the doctors are using is, as Arielle said, is calprotectin. And that's not a very precise biomarker, although it's measured because it's indicative for telling the doctors if there is inflammation in the gut.
Mm-hmm. So would you say disease signature helps us figure out something like precision medicine, which is?
So precision meaning like precise, right? Also can be used synonymously with like personalized medicine. So I think the best like example of that is, really cool like immunotherapies for cancer. So they'll actually like T cell therapies, they'll actually pull out your own T cells, which are a component of your immune system. They'll modify them genetically, and then they'll put them back inside of you. So that's like the most personalized type of therapy ever, because it's your own body is being re-engineered to fight something, right? And so right now, like a personalized medicine approach in IBD would be something like, instead of just like throwing a drug at you because you fall into this bucket of like mild to moderate UC, we would try to narrow down more specifically what type of drug would be best suited for you, given all of these different disease signatures that we're picking up. And it's a much more, I think you can think of it as more targeted. It's a more targeted, personalized approach as opposed to just bucketing everyone in like two different categories and then trying medications on them.
So, for example, the current precisions is not very precise in diagnosing IBD. Then one of the main problems, we actually talked, this Friday to one of our IBD consultant advisor, and he said that, one of the main problems, the precision to diagnose ulcerative colitis and Crohn's are not very precise. It's about 70 to 80% precision, which means that a person that have ulcerative colitis may have Crohn, but it will be diagnosed as ulcerative colitis. And this is really important because if you get to the higher precision, you will be able to treat the patient more effectively with appropriate medicine and appropriate management.
And quicker, which we also know is super important, right? So like getting your disease under control as soon as possible, as opposed to like waiting and trying out a bunch of different medications and then hoping you find the right one soon.
A couple years ago, I was in a Starbucks in Chicago talking to our friend Dr. Nandi about precision medicine, and he just like blew my mind all over my face when he was really talking about this. And I was like just thinking about the different outcomes that are possible once we finally figure all this out. Like it's mostly a concept right now, but once we get there, just thinking about the patient journey and how it will significantly differ from mine and other people who are currently diagnosed or newly diagnosed right now. And it's super, super exciting. We'll have to do a chat about that another day. One more term that I'd like you to help me with, genomics.
Mm-hmm. Yeah. Arie, take it away.
Genomics, that's a good word. It's a name for the entire understanding of genes associated with any organism. So, for example, everybody is made of genes at every cells, and the activity of these genes in each cell or with the context of the disease are very important for understanding. So the studies of the genes functions in normal person or the disease, and the inquiry about understanding the regulation of the genes are called the studies of genomics. So applying genomics to diseases like IBD could provide a lot of information about the genes function, genes activity associated with the disease.
Yeah. And also importantly, it's not like looking at one gene. It's like you're sequencing like the entire genome, which just like a couple years ago was like crazy. And now it's so easy for us to do. So I think like the technological advances that have happened with sequencing and being able to like actually look at the whole genome has really opened up such an interesting therapeutic potential within IBD especially.
So all this talk about, precision medicine and genomics and all these things kind of brings me to the next question, which is, historically and currently, IBD is really difficult to classify, to diagnose, to treat. Arielle, can you talk a little bit more about like why it's so hard for us to figure this out in the current present life we're living right now?
Yeah. So I think this stems from kind of like it's really easy. I think within medicine, we want to like slap on diagnoses on people. So you kind of want to put on labels, right? And it's funny because IBD is like inflammatory bowel diseases. So it's this like umbrella term. And then within that, you have predominantly ulcerative colitis, Crohn's disease, and what they call indeterminate colitis. And even within those, like just within Crohn's, it's totally dependent on which location of your intestines you have affected by the illness, as well as within UC. And then you also have severity. And so there's all of these like subtypes. And so what Arie was saying, where it's like sometimes you'll get diagnosed with UC, but it's actually Crohn's disease that was just limited to the large intestine at the time. And you'll hear these really sad stories of people having total colectomies because they think it's going to be like curative of their UC, and then the Crohn's will actually pop up somewhere else in the small intestines. So it's really hard to treat because we just don't actually have really precise ways of diagnosing. And I think I'm hoping that taking this more of like a precision medicine approach of actually trying to understand these individual disease signatures will better classify the disease across the different types of patients. Because right now, the diagnostic terminology isn't really mapping onto the experience very well.
So it believes that IBD is probably like a lot of other diseases. It's a spectrum disease, which means it's more than 15 to 20 different diseases. And this disease is treated basically on the symptoms, not on the cause of each disease. So imagine that if you would be able to precisely figure out each of the cause of each symptoms associated with 20 different diseases for IBD, and have a targeted therapy for each of these particular causes, you have a much more precision medicine, much more precise treatment, and more effective treatment for the patients. So.
Mm-hmm. Yeah, it feels like we're so close, right? We need to first figure out how to classify, then we can help with precision medicine. I mean, we're so close, but so far. But really, all of the pathways, the networking, it's there. The plan is there. We just haven't figured out a way to like get the plan to happen yet.
Well, we're working on that plan. Plug for the next question.
The map is there, but how to navigate it is the hardest part now. So.
Well, speaking of navigating the map, back to you, Arielle. Let's talk about IBD Footprints program that Gali Health is running and hosting. We talked a little bit in the beginning about the differences in research, but why is this program different than anything else that's happening right now? Like, what is it? The study itself? Is it the approach? Like, what's different?
I think there's a lot of different things. My favorite thing that makes us different is the fact that we embrace this problematic heterogeneity that we were just talking about. So a lot of research studies within the space try to reduce like what we call the noise in order to get a signal, right? because like there is so much heterogeneity within the illness that it's really difficult to like pick up on any of these potential signatures or biomarkers when you have such variability in the experience and then the way that the disease is manifesting. So for us, instead, we're taking this like huge kind of like broad approach where we're actually trying to get pretty much everyone who has IBD enrolled in our study within the United States as well as the world. So we actually just launched in Canada, which is really exciting. And we're going to be going global very soon beyond North America. That is the goal. But the only inclusion criteria for our study is that you have a confirmed diagnosis of IBD and that you are at least 18 years old. So that means if you have a J pouch, if you have a colostomy, if you have, if you've had the disease for 20 years, if you've just diagnosed, if you've never been on a biologic, if you've exhausted all of the biologics, we want to have like complete representation in our study because it's going to take us, we believe that it will take us embracing that heterogeneity in order to fully characterize the illness in the precise way that we've been talking about. So I think that's one of the things that I really love is just how inclusive we are. And we're actually trying to characterize the diseased population in the way that it actually exists. The other thing that contributes to that is it's a completely remote operation. So we were talking about how like this is completely, 50% of the program is actually just giving us, telling Gali how you're feeling every day. So as using, normally if you have the Gali app, you know that if you just fill out your health and treatment monitors every day and you fill out your surveys, she ends up personalizing your health feed with all this amazing content that's relevant to you today. That's pretty much like half of the study already. So if you're already doing that, you're almost there. The only difference when you're participating in Footprints is that we layer on these, what we're calling biosamples. So we collect stool samples and we also are collecting saliva samples. So what I think is really cool about this is that traditionally, if you wanted to participate in research, you have to live close to an academic medical center, which within our program, you don't have to at all because we send you everything you need at home. So all you need is a smartphone. We're about to launch Android actually. So even if you don't have an iPhone, that's not a problem anymore. You just need some kind of smartphone to be able to tell Gali how you're feeling every day. And then we send you kits to your home to collect stool and saliva samples. So I think that also helps us increase the representation of the patient population because we can get people from all over the country, especially in rural areas where typically, those people who live in rural areas or away from like major academic medical centers have a hard time getting involved in research. So I think that's one of the cool things about it.
It's also worth noting that I just got my reminder to fill out my daily monitor while we're talking right now.
There you go.
You get reminders daily to do it. And I know you mentioned like it comes to you, but the post office will also come to your house to pick up the sample when you have to send it in. So you literally don't have to leave your house. And for me, as somebody with a J pouch, one of the things that made me most excited about Footprints when you guys first told me about it was that I could participate because like you mentioned, people with J pouches and ostomies are often excluded from clinical research. I mean, for good reason when they're testing certain things, but it's almost like we don't matter in terms of research anymore. And all of us still have IBD. So the experience does matter. So that's really cool. And I'm really, really excited about that part of it too.
I want to add something to Arielle. So, I mean, the reason we're very excited about what we're doing is, a lot of you probably have seen a lot of studies in academic centers that they did a lot of genomics and studies of all sorts of patients and reported all sorts of signature, and none of this, they generated really sexy papers, but nothing really turned out to benefit for the patient. And one of the reasons that we feel that our approach is different, we're not simply going to do a snapshot analysis of the patients. Because our technologies allow us to do self-collecting the samples of the stool and analyze them over a long period of time, tracking the changes over a long period of time would allow us to really understand the dynamic changes over a course of the disease. And no one ever done anything like that, to our knowledge, that follow the patient in a long period of time and not just took a snapshot. And the reason is because most of the analysis, the genomics or the markers or any of the molecular analysis that were done in the past were done on a biopsy samples where you have to go and get a colonoscopy, and you could not really do this on a regular basis. So our technology allows to really track these activities over a long time, and it's most likely going to really spore and identify important changes in the patients.
Yeah,
So
that's true.
let's talk a little bit about that technology and get right into the sample itself. So we didn't talk at all really about like how the sample is collected, but I'll just tell you it's super easy. It's not hard. It's just like easiest thing ever. But so I've sampled, I send it into the mail. Where does it go? Who handles it? Like what happens when you get my box of poop? What do you do with it?
So it's really interesting because I think that there are components of the kit that will make sense after we talk about this. So you'll notice that in your tube, so whenever we give you everything you need to collect, right? And so there's a little tube that comes with, you have a toilet hat, so you can go to the bathroom, you can collect your sample. There's a tube and the cap has like a little spoon on it, like a little spatula. It's super easy. You just scoop, put it in the tube, send it out in the envelope, prepaid, whatever, super easy. In that tube, you'll notice that there's liquid. And that liquid is really what empowers us to be able to do these at-home collections. So that liquid is what we call a stabilizer or a buffer. And essentially what it does is it completely snaps. It's like a snapshot of everything that's going on in your sample at that moment. It completely stops all of these processes. So for instance, with like bacteria, we know that bacteria is constantly propagating and changing, and they're, doing all kinds of things. And especially once the environment changes. So once the stool with your bacteria is outside of your GI tract and it's in this new environment, the microbes inside of it are going to start doing all kinds of different things. But once you put it in that buffer, it actually like stops all the processes in time. So we get this really great snapshot of what was going on inside of your GI tract right when you collected. So that's the first thing. The other thing that's cool about it is it completely renders the sample like non-biohazard. Like it basically makes it so that you can easily ship it without it being like, is it okay that I'm putting poop in the mail? So that's not to worry at all. And it also keeps the sample from degrading or having any problems for like a full 7 days. So that's what allows us to actually collect a sample at home without you having to put it in the freezer or immediately dropping it off. I'm sure people have done stool samples for their GIs in the past where you have to like get it to the clinic very quickly because it's not in stabilizer, right? Okay, so it's in the stabilizer, it's in the mail truck going off. It goes to a biobank. So a biobank is like a regular bank, but instead of money, it's a bunch of biosamples. And it's like a shared resource that a bunch of different labs use. And it's just full of different freezers, essentially. You can think of it as like a warehouse for your biosamples. And so for us, what that means is that your tube goes to the biobank. And once it gets there, every time you like thaw a sample, which I think is really interesting, it actually degrades the sample. So let's say that we put this whole tube into the freezer and it's like minus 80 degrees. It's very, very cold. So it can stay there for a very long time. So that way we can like look at it in the future. If we, discover any new technologies that we want to be able to use on your sample, we could. But every time you pull it out of the freezer and it thaws, it actually hurts the sample. It makes it harder for us to detect things in it. So the first thing that they'll do is they'll create these things, a jargon term called an aliquot, which is basically, you can think it's like a smaller sample. It's like taking your big sample, your big tube, and making it into a bunch of little tubes. So that way, every time you want to do something with it, you only have to thaw it once because now you have multiple tubes. So you don't have to have this one big tube that you're pulling out of the freezer, taking a little bit out of it, putting it back in the freezer. And then if you want to use it again, you've already gone through a freeze thaw. So essentially that's what happens.
What was that term again?
An aliquot.
I feel like I need to start using it like as an insult. It sounds like a great
For an aliquot.
Yeah, it's like a great, it sounds like really insulting.
Yeah, in our case, it's a little poop, essentially.
I mean, kind of the same, right?
Yeah. Yeah. So that's basically like the first thing that happens. And then once we're ready, once we have enough samples, we'll take one aliquot of each person's sample and we'll extract DNA and RNA and other things from it and do different types of what we call assays, which is treatment of the sample to detect different biomarkers that we're interested in.
Can you go over what RNA is?
Arie, take it away.
Great.
This is on you.
Biology 101. Okay.
Let's go.
Okay, so for those of you that never heard about RNA, and just a really brief thing, this is a good thing to know, just not for IBD. Every cell has all the genes stored in the nucleus as a DNA. The DNA is copied, a gene is copied and become an RNA. And the RNA goes out of the nucleus to make a protein, a specific protein, as the blue script of the DNA is telling it. And the protein does all of the function of life. So when one wants to really understand what is the activity of a particular cell, one looks at the RNA population in the cells, and that's indicating what is activity currently happening in the cell. This is so important because in a disease state, what happens, certain genes are making more RNA of a particular one. So really knowing which RNA is made, excessively is indicative about the disease state, the disease signature. So we want to capture this activity and infer from that information what's happening in terms of the activity of the disease.
Yep, exactly. It's really the main difference between DNA and RNA is like, just because you have the DNA of a specific, just because you have a specific gene doesn't mean it's actually active or doing anything at that moment. And so if you really want to know like what the activity is, then you would want to look at RNA because that's actually what then creates or results in proteins being created, which are the actual chemical messengers that do things.
Okay, so this is so fascinating, you guys. So we send in the sample, you start looking at the sample. Arie, can you tell us like what you're looking for in those samples? I'm gonna guess it's something to do with RNA, but
It's RNA. So there is three things that are most important in IBD. Okay, so a lot of you probably know that IBD is a disease associated with the breakage of the mucosa, which is the lining of the gut. And because of the breakage of the mucosa, there is exposure of the inside of the body to the microbiome, to the bacteria that resides normally in the gut. That's often causing chronic inflammation, which means a lot of immune cells coming to that site and try to fight these infections. And because of that, some patients have a chronic inflammation and they cannot clear the inflammation. That's the reason most of the drugs for IBD are anti-inflammatory drugs. So the interaction between the microbiome, the bacteria or the microbes in the gut, the immune system that comes to protect the body in the area of the inflammations, and the biology that associated with the mucosa, with the lining of the gut, is very important. As you can imagine, it's a war zone. The immune system is referred to like the army. The army comes in and try to clear it out. And sometimes the army has a friendly fire and is super active. And one needs to really calm the army down to really, stop the war. So that's where the anti-inflammatory drugs. The other area that we're looking for is genes activity associated with mucosa biology. What are the genes? Are there any defect in the genes? Why the lining of the gut breaks so easily in IBD patients? Why it could not be repaired easily? So there is a lot of information at the level of the RNA or the DNA from these cells. And the third interface is, of course, the microbes. There is a certain, although the microbes that lives in the gut, in 99% of them are friendly and important for the maintenance of our GI, but some of them becomes active during inflammation in IBD. So understanding which bacteria or which microbiome is active in response to the inflammation or participate in inflammation is very important. So we capture all of these components, analyzing them, and try to find what is the relationship between each others. I don't know, is that clear? Too scientific? I hope not.
I don't think it's too scientific. It's just like, whoa, so much information in a really awesome way. But that's why we're doing this though, right? Like I wanted to know when I participated, like, what are we looking for? I mean, another question related to is when you first submit, you submit a saliva sample and a stool sample. And are you looking for the same things in both of those samples?
Yes, the saliva has microbiome too. There's a lot of microbes in the saliva, and there's a lot of information about immune cells associated with saliva. I would say, for IBD, the most important sample is a stool, because that's the site of the injury. And, in the site of injury is going to be a lot of information. And based on the, in the injury site, you can really see who are the participants in this war zone, and try to understand why are they this participant there, and what their contribution for the disease. So once you understand those participants, you can really figure out, diagnostic, you could figure out therapeutics, and you could figure out better management for the patients.
Fascinating. So we send in the stool sample and you guys get all the fun data and microbiomes and all the other cool jargony words that we talked about and stuff. And then we self-report stuff in the app itself. So Arielle, can you tell us how those two pieces of data kind of work together to form the study, and why the study that's why it's unique, right? Is because these two things work together.
Yeah, so if the biology wasn't complicated enough, right? We also know that people can experience inflammation in all of these processes in their gut, but be asymptomatic. And we also know that people can be completely symptomatic and have absolutely no evidence of inflammation in their gut. So this is very weird, right? And we don't really know what the differences between those are. And if you're experiencing all these symptoms, but there's nothing, biologically showing anything according to your colonoscopy, what does that mean? So I think for us, we understand that the patient experience and what you are experiencing on a symptom level, your lifestyle, everything, every single day is just as important as what's going on with you biologically. And we feel that the only way for us to really kind of get these disease signatures is to incorporate what I was talking about before, these digital markers. So there's biological markers, which is what Arie was just talking about. And there are also these digital markers, which include the PROs, the patient-reported outcomes. It also includes things like we ask about how stressed you're feeling. We ask about how much sleep you got last night. Did you get good sleep? We ask how physically active you're being. We're also looking at kind of what you're doing in the app, what types of information are you looking at? What are you interested in? Because we know that, patients really are experts of their own disease. And so whatever you are going through each and every day as part of being in the study is just as relevant as important as what's going on with you biologically.
So I'll give you a quick example that we talk to the people. So for example, one of the most common drugs for IBD are Remicade and Humira. And those antibodies were developed to block immune regulator that regulate the immune system. It's called TNF-alpha. So if we are tracking the stool of these patients and we seeing that a given patient is on one of these drugs, and that patient is not responding to the drug, as a large number, more frequent flare-up incidents, and we potentially could really zoom in and see that the level of this particular immunoregulator is changing, it would indicate that this is the wrong drug for those patients. So it gives some, so the coordination and the association between the patient reporting symptoms to the treatment, to the molecular analysis, could really influence a decision of the treatment for these patients.
So because we're not quite at precision medicine yet, this data that we're submitting, whether it be through stool, saliva, or digital biomarkers, can a participant in Footprints see that data? Can they use it? If so, what can they do with that data? Arielle?
Yeah, totally. So I think I want to make it very clear that we cannot draw any clinical conclusions from anything we provide. Exactly for that reason, right? Because we're just not there yet. We're still in this discovery process. But we also fully believe that it's important to be able to give back because patients are giving us so much as part of this program that we want to provide some insights for them around what it is that they are submitting and the things that we're learning about them. So it is our goal, and this is part of the consent form, so we're very explicit about this, that we want to be able to provide participants at least one microbiome report of one of their samples within like 2 to 3 months after their 4th sample. So of course, due to COVID and everything that's been going on, we're a bit delayed in doing our assays. So that's why we ask for that buffer period of like 2 to 3 months. Once our systems are in place, I think we'll be able to get quicker with this. But we do really want to be able to provide a microbiome report, especially because I know that people are just intrinsically interested in like, what are the bugs in my GI tract? Like I'm interested. I think everybody, like the microbiome's so hot right now, just like even just knowing what's in there is fascinating. So we're trying to figure out what the best way of actually presenting that information, because again, it is so complicated. But that is our goal. So you will get to see that. And also, as you know, like using the Gali app, you get regular check-in reports every 2 weeks of your data. So every time you're filling out your monitors as part of the study or just as part of using the Gali app, you're getting these really cool check-in reports every 2 weeks that show you trends over time, you know, significant associations between different symptoms and lifestyle. Like, oh, like I've noticed, you know, Gali will say, like, I've noticed that on days that you're not sleeping well, you're also reporting more abdominal pain. So that's all a part of the Gali app experience in general that you would get. So that's part of it, but also you would get your microbiome report for one of your samples at the very least.
So with all the data that's being provided, can we talk a little bit about data and data privacy and protecting participants' data? Because that's also a big hot issue right now. So what is Gali doing to protect the participants' data?
Yes, so really cool. When you get, I don't know if you've noticed, but whenever you get your kit, you'll notice that it's labeled with some letters and numbers on the tube. So that is what we call de-identifying your data, right? So there is a secure, like an encrypted database that really only like me and like members of the research team have access to, that links your personal health information or identifiable information like your name, the address you send your kits to, things like that, to that little like letter and number code that's on your tubes. So none of your data, like anything around like the RNA that we're looking at, anything in terms of like what's going on with your monitors, none of that is connected to your name or anything that would identify you. So it's all like de-identified is the term we use for that. So that's following like good clinical practices for human subjects research. The other thing that's important is that even though we're going to be giving you these microbiome reports that like show your individual microbiome analyses, anything that we're looking at is in aggregate, meaning that we're looking at groups of people. So we're combining a bunch of data from a bunch of different people and looking at it in aggregate, meaning that like we would never like say like, hey, here's Mary's, you know, microbiome to anyone else. Like that would never happen. So anything that we're doing in terms of like publishing findings or sharing with the scientific community, things like that would never involve just your data alone. It would be in summary with everybody else's data. So take home is that you will never be able to be identified by your poop or by the data you put in the app.
Yeah. What a relief, right?
Yeah, don't worry. Don't worry.
We have a question from our viewers, which is just how long is the Footprints study?
Totally, yeah. So that's another thing that I think is really cool. So it is also personalized because we recognize that like some people might be up for more than others. So we're actually currently working on a tier system that will be coming shortly, hopefully soon. But for right now, you can either sign up for 4 samples over 4 months. So you just sample 1 time a month. Like you said, Gali reaches out and reminds you. And if you don't respond to her, then I reach out and remind you. So you'll be hearing from me. The poop collector. So you can do 4 months, and then at the end of that, if you're down to do more, you can do it for up to 12 months. So you can do it up to 8 more times. So it's completely up to you. There have been people who are like, sign me up for 12 at the get-go, which I'm loving how eager people have been. But if you're like, I don't know about that, that seems like a big commitment, you can always just sign up for 4. So it really just comes down to collecting a sample once a month, and you just have to do your health and treatment monitors at least 2 times each week while you're in the study. Because again, if we don't get that monitoring data, then we don't have any of the patient-reported outcomes to connect to your samples. So those are the 2, that's the commitment of being in the study.
Got it, got it. Okay, so footprints overall, right? The end goal. What would you say that the end goal is? And I'm gonna add an and onto that and direct it towards Arie, which is, what biomarkers are we looking for? And is that the end goal, is to help find new biomarkers?
So we're looking for biomarkers that would allow us to classify patients better. So if we could really increase the precision and diagnosis for Crohn's to UC in a 90 or above precision, would be very happy to bring it to doctors or to whoever wants to use it in the future. And the other area that we are very excited and thinking very deeply, we talk to a lot of doctors and try to understand the space and what are the unmet medical needs. And one of the unmet medical needs is to find a molecular signature or biomarker that could really help to the doctor to match the right therapy to the right patients from the get-go. If the patient is already, although it could be that the symptoms manifest themselves not severe, but the biomarkers suggest that this is a very advanced disease, that would suggest to the doctor to use an aggressive therapy from the get-go rather than let the disease progress to something more severe where the treatment would be much harder in the future. So understanding the disease severity and matching the right therapy with the right patient, this is the ultimate goal.
Arielle, what do you think the end goal of Footprints is?
Yeah, I mean, I think I totally echo what Arie said. And the only thing I would add to that is, like, in the end, like, all of this is really making it so that it's easier for people to have IBD. Like, that's really what it is, because I feel like, you know, it's a pity that like so much advancement has happened in the therapeutics realm. Like, we've talked about how, like, back in the day, like, these drugs were not available. Like, but the age of biologics is this like whole new thing, right? And it's really changed the game for a lot of patients. But now that there are so many different medications that we don't actually know which ones are the best for different types of people. And so I think if we could now take what we've learned around the mechanisms and actually match them to the appropriate people at the right time, then I think that the disease would become much more manageable. And so just essentially, really, it's for us, it's, you know, it'd be really cool to just, like, from a scientific standpoint, figure out these mechanisms, understand the biomarkers. But if it doesn't really map onto clinical decision making and making patients' lives easier and more manageable, then, like, there's really no point. So I think that's really the end goal. And so for helping physicians, you know, manage their patients and help them make treatment decisions together, I think ultimately that just makes lives easier when you're diagnosed with this illness.
And you at home can be a part of this by just sending your poop in the mail, right?
Yeah,
like,
pretty much, yeah.
That's all you gotta do. We can just get right to the end. I mean, because that's what participant samples are doing, right? Sending in your sample means you're participating in this research where we're looking for all this stuff to make precision medicine happen, to make our lives better, to make everything go smoother and be more awesome for people in the future, right?
Yeah. And hopefully, like, our goal, we're really aggressive around this. Like, when we say the future, we mean like the near future. Like, I think oftentimes we're, especially in academic research, we're like, this probably won't benefit you, but it will benefit like people in the future, some undisclosed period of time, who knows. But I think for us, especially where technology is today, we want it to benefit you. And so for us, that's why it's so important that everybody and their different experiences participate, so that whatever we end up finding actually does work for you. So I really encourage people, regardless of your experience, it's such an easy thing to participate in. you're helping your community, you're helping the scientific community, you're helping your GIs, and like hopefully you're helping yourselves, and you get to find out what's in your microbiome. So that's pretty cool too.
Hey, speaking of microbiome, we have a question from somebody viewing, which says, you know, like, what can we do with that microbiome report?
Honestly, so I think for us, like again, clinically cannot make any recommendations. I want to be so clear about that. I do know that certain people are really interested in like what specific microbes do. So kind of like what we were talking about with RNA, right? Like what is the activity? What is the function? What are the different like metabolites or proteins that these microbes are producing? So like short chain fatty acids, like they're actually, you know, all like most of the serotonin in your body is actually produced and metabolized by microbes in your gut. So learning about the different types of microbes and what they're doing in your GI tract could inform, like, I don't know, maybe choosing to eat a specific food, or we would never actually make those recommendations to you. But if you were to go to your physician and be like, hey, like this is something I'm learning about myself, do you know anything about this? You're totally available to do that. But Gali Health we're just not at the place scientifically to like actually make any kind of clinical recommendations around what we find. But you can totally go and learn all about what these microbes do. And we hope to at least provide educational information around what different things mean around what's inside your gut.
And if nothing else, you can just nerd out
Yes.
into perpetuity with your own data. Like how
Yeah, frame it, put it on the wall,
right?
and say microbiome.
I was
What's your
gonna
microbiome?
say, so microbiome reports, like the graphs that you end up getting are so beautiful and colorful that I was thinking like, we need to turn these into Instagram posts because they're just so cool.
Yes.
So my goal is to make your report like very social media fun so that you can just like share it and be like, welcome to my microbiome, and brag about the microbes.
It's such a millennial Gen Z thing to say to it, and I'm here for it, I have to say.
That's what I would.
We had there's going to be potential information there, but it's not. It's beyond our scope. Is for fecal treatment. There is a lot of this kind of therapies, emerging therapies. So if you have a particular microbiome, potentially one could really see if you're a candidate for that kind of a treatment. So if you get a picture of it, and if they're engaged with companies or organizations that are doing this kind of treatment, you may bring this map and show it to people. Say, hey, this is what. So I don't know, but that could be useful.
Ugh, the inner nerd in me is so excited right now. So I am gonna wrap us up right now and just say thank you both for joining me. Thank you for dropping all the knowledge bombs on the IBD Footprints program. Like I said, I'm a part of it. It's super easy, and even when it's not easy, Arielle will help you remember how easy it is and send you friendly reminders when you forget. And Arielle obviously is super cool, so it's like no pressure even when you forget. But yes, thank you both so much. And also, really quick, Arielle, if somebody wants to join IBD Footprints, what do they do?
I was gonna say I need to plug how to get in. So all you need to do is like download the Gali app. The easiest way to do it is download the Gali app, tell her I want to join the IBD Footprints program by just sending her a chat, and she'll get you all set up, and then she'll let me know to send you a consent form. So she's really easy to work with on that. Or you can always email ibdfootprints@galihealth.com if you want more information. You can also check out our website, galihealth.com/ibd-footprints. That has everything about it. There's lots of ways to get in touch. Email me, Arielle@galihealth.com. I'm happy to talk more about it if you're on the fence. Any way works.
Lovely. All barriers removed, no excuses, right?
Exactly.
All right, thank you both again. Have a great night and go home and join Footprints if you're listening.
Yes. Thank you.
Bye.